So going back to treatment of psoriasis, currently, of course, we're not using sun. Oh, we might use sunlight sometimes in very sunny areas, of course, as well as Italy, of course. But of course, the mainstream of the treatment for psoriasis is very, very rich and populated by several different drugs. So all the data that we are studying, we are reading, comes originally from randomized clinical trials. And when you recruit patients, some of you might have participated, of course, in randomized clinical trials. When you recruit a patient, it's very clear, it's very clean, should be a clean patient. Severe psoriasis, a lot of exclusion criteria. So it's a very direct and straightforward path. It's like going into a train with just one rail. Instead, real life is way different. You have several directions, several rails to take, and of course you can have several variables impacting on the choice of the treatment and the use of a specific drug instead of one other. So previous treatment, infections, comorbidities, and so on. So that's why real world data are quite important currently, compared with what we used to have basically in the past. We only relied on RCT back in the past, but now, of course, we rely still on RCT, but we are providing more and more data about real world evidence, real world data, real life, let's say. So there is a very different scenario here. So a randomized clinical trial is asking, can the drug work in a highly selective, specific, homogeneous population? And real-world data are exploring the effectiveness of a drug in a very complex, heterogeneous group of patients. So it's basically what we face every day. So elderly patients, pregnant ladies, and so on. So this is quite a big difference. And of course, even subtle differences in study design can impact the absolute effect of a treatment. So basically, clinical trial settings are very clean, very direct, let's say, comparing 2 drugs or even 3 drugs sometimes and so on. But what we are doing in real world setting, we are comparing several drugs, maybe we are comparing, maybe retrospectively, most commonly, a lot of drugs in our databases, in our experience, and so on. The sources of real-world data are really various and can go from patient registries to your own database that you have in your clinic, in your hospital, and so on, or even electronic health records, specifically in big countries where they collect data about health records, specifically, namely for insurance purposes. So, of course, real-world data are currently seen as quite important, even for payers or even for major organizations like FDA or EMA. Why? Because they can support data from clinical trial. They can give new data about, of course, new, let's say, questions, new outcomes that you want to explore after the RCT is already being published and established. So it's quite important to have real-world data currently, and real-world evidence comes from real-world data. So when you refer to RWE, you mean evidence, but of course, it relies on RWD. That means real-world data, of course. This is just to be clear here. So what do we do with real-world evidence? Of course, we need to be sure that the data are well collected and potentially and hopefully are gathered from prospective patient registries or prospective cohort studies and so on. Those data, the prospective data, of course, are way they tend to be way more accurate and complete than the retrospective collection of data. Unfortunately, I would say 90% of real-world evidence comes from retrospective database or administrative claim records, insurance claim records, or electronic data banks, especially in the US. So there are quite a few differences here between RCT and RWE that are already well known, I guess, by most of you, and are crucial if you want to compare data. Maybe sometimes you can do comparing real-world evidence to with randomized clinical trial data. So first of all, we define efficacy coming from RCT and effectiveness coming from real-world evidence. It's just a matter of saying different things, but indicating the same goal, aim. So for instance, in psoriasis, the clearance of psoriasis or the achievement of PASI 90 or whatever you want. So in the real world, you call it effectiveness. In the control environment, you call it efficacy. And what's more, when you use this word, this meaning here, this wording here, efficiency, you also introduce another topic that is the cost, or let's say the overall worth, let's say cost and generally speaking balance, let's say between cost, effectiveness, or the not only the cost but also the burden, let's say the indirect cost maybe sometimes of the drug related to the drug compared with the effectiveness. So if you say efficiency, you mention also something related to the cost. So when would you yourself perform a real world data study, or you would read about this? But well, you can basically provide data from the very beginning, even before the launch. This is quite uncommon, let's say. It has been done by several investigators lately, even in Italy, but it's not very common because normally you perform a real world data study, of course, after the introduction of the drug in the market. Then specifically when you want to understand if the price is correct or is worthy, so you make something related to efficiency, well, then you use a lot of real world evidence to provide data and to sustain the use of one drug or the other. Of course, when you have new competitors, you do a head-to-head in real world evidence, and of course, you might target a subpopulation that has never been treated, for instance, in randomized clinical trials, namely with hepatitis, active hepatitis, and so on. So you see, it's quite complex. Let's say the use of real world data in this scenario. Let's go on and try to understand why real world evidence currently has been considered so important and received so much attention. But well, for instance, pandemic as COVID pandemic, of course, has literally made us think about what we could do. Well, everybody at that time was providing data about COVID and patient under treatment for psoriasis, trying to understand, for instance, if biologic were detrimental or even positive in the outcome of COVID itself. So generally speaking, COVID with vaccination and with new drugs and so on has made everybody understand that we really need to rely on real world evidence, because clinical trials, randomized clinical trials, are too difficult to perform in a rapid time. They need a lot of time, they need a lot of investment. So therefore, real world evidence is much more important, has been considered much more important even after a pandemic. But stop for 1 second, let's stop to think about this and to think about the potential problem we face with real world evidence. Let's speak about COVID. I just mentioned COVID. So I myself, with a lot of colleagues, Antonio Costanzo, Paolo Gismondi from North Italy, we collected data and we published a lot of papers in important journals, and very rapidly, I must say, it was the fastest publishing period of our life, I guess. And we were collecting data, of course, about the impact of biologics, specifically in the COVID-19 outcome in patients with psoriasis. And we were, after a bit, understanding that a lot of people were publishing different results, and the inconsistency, the unhomogeneity of all these results made us think that maybe something was not correct there. And we also published this. We collected data. We analyzed the literature after a couple of years of the pandemic, saying something like this, that probably the need to rapidly spread information to the dermatologic community may have hampered the methodological rigor in the published literature. So we analyzed the published literature, finding at the end 23 studies meeting the criteria. And for them, the median NOS, so the Newcastle-Ottawa scale score, was rather low, was less than 5, 0.5, 50%, whatever you want to call it. When it's less than 7, it is highly biased. So basically, the quality, the general quality of the published literature at that time was not really high. And of course, this is, you might be familiar with the Newcastle-Ottawa Quality Assessment Scale for cohort study or for case control study. And it is a good way of judging the quality of the observational study. So despite this caveat that I just mentioned, of course, real-world evidence is transforming healthcare now. So FDA regulators, generally speaking, are currently analyzing even real-world evidence, not only clinical trials, randomized clinical trials. And of course, it provides opportunity to find new data, new findings about specific subpopulation, or for instance, safety-wise, to identify rare safety events that is not, of course, feasible during, let's say, a low number population, a randomized clinical trial. So this is quite important, but we still need to be cautious. And talking about strength and limitation about all these studies that we can perform, we can publish, of course, we already been published a lot of real world data studies. So strength of observational studies are a lot. We are fast recruiting this patient, especially retrospectively. We are very, very good in collecting data, maybe from different centers. That can be, of course, a plus, but that can also limit the analysis because all the centers might have different approaches to patients and so on. So those limitations are very important. And I will skip this for a matter of time, but I will show you a couple of examples of how you can perform good, really real world data studies. For instance, there are also companies, big pharma, working on this. This is a Lilly study, but of course, also Johnson & Johnson performed recently a lot of real-world data study, both in PSO and PSA. So for instance, this is just an example. SOHO is a prospective study again, so that's the highest level of data you can provide in your study, of course, and comparing different biologics, recruiting a lot of patients in a lot of countries. And just to let you know something, so when you recruit this patient and you don't select them, you don't blindly select them, you don't perform any a priori, let's say, assumption, you might find that your population treated with different drugs is quite different. So, for instance, there are quite relevant differences in this SOHO study, even if it's prospective and supported by a big pharma. So, for instance, let's check the psoriatic arthritis patients. So only 12% have been treated with risankizumab, compared with, for instance, 27% or 30% treated with IL-17 inhibitors. So this is of course, what we call channeling bias. I will focus on this later. This is just result from SOUL study. Also, we were able to focus on specific sites, special area. That's very, very easy when you do a real world data study in this case, for instance. Again, let's go back to weaknesses or threats even about this real world evidence. So the strengths we already mentioned a couple of times. Weaknesses, we already mentioned this, biases and confounding factors. I will focus in the coming 5 minutes on this. And of course, threats, because you might think it's all safe, but sometimes, especially if you collect data retrospectively and so on, but sometimes you have ethical concerns. You need, of course, informed consent. You need to go to your ethical committee, institution, local or regional or national, and so on. So statistics is the only science that enables different experts using the same figures to draw different conclusions. For instance, this nice paper compared studies from real-world data, real life, let's say, and real randomized clinical trials. Studies assessing exactly the same outcome, recruiting kind of the same patients. So basically, this is RCT in the x-axis, in the y-axis is the observational studies. So there are very, very evident differences in some studies showing that you might have a different hazard ratio or odds ratio, quite different, even extremely different in some of the trials, some of the observational studies. That, of course, raises some concern. And why this is happening? Because you will face biases when you perform an observational study. Let's see a couple of examples here. And let's mention a couple of biases. Probably the strongest, the most common is confounding by indication. So when you compare different drugs in a real life data, you for sure needs to consider that you prescribe, generally speaking, you prescribe a drug A for a specific type of patient in your mind and drug B for another type of patient. And you need to control for these differences. And when you control everything, it's all fine. When you do a prospective study, generally speaking, you try to control for everything. But when you do a retrospective study, or you recruit data from, I don't know, registries or databases and so on, you will not find all the potential confounders, all the covariates that can eventually impact on the outcome. And another bias, very common, is channeling bias. It's something similar to confounding by indication. So when you try to prescribe, or you tend to prescribe a specific drug to a specific patient. So another big problem with real-world evidence is missing data. Of course, we already mentioned we might have a retrospective study, might have registries or electronic registries without a lot of covariates that can eventually impact in your analysis. And another very important bias is related to time. And for instance, when you compare, for instance, biologics with conventional drugs, you know for sure that biologics are coming later in the history of psoriasis compared to the traditional drug. That could, of course, introduce a bias when you compare these two cohorts. And how do you handle these confounders, these differences? Well, you can do a lot of things. You can do multiple testing. So when you do multiple sets testing, you need to correct for multiple state testing, for instance, with Bolferoni tests. You can do sensitivity analysis. You can do something that I will mention right now in this case study. It is a very important topic. I have still 5 minutes to cover this. And this, I like this topic because it's very important. It's very hard. Is how to stop the progression of the disease, or how to intercept or prevent the development of arthritis in psoriatic patients. So you have a lot of studies there, and you will find a lot of, let's say, contradictory results. some studies actually show that use of biologics rises the the risk of development of PSA in your patient with psoriasis, paradoxically, I would say. So, for instance, this one was one of the first paper, probably the first paper from my friend Paolo Gisoli from Verona. And interestingly, when they showed the figure, they showed that biologic treated patients were developing less PSA than patients treated with phototherapy. However, when the reviewer asked them to perform a propensity score matching, Paolo told me, Professor Giannoli told me that actually the hazard ratio was split, was completely the opposite, showing that actually the risk of PSA was higher with biologics than the phototherapy treated patients. That's something, of course, we need to consider. Propensity score matching is a powerful, that's another example. I will go back to the propensity score matching, sorry. This is another example from Mir. This is an American study. Joe Merola is there too, Gelfand, OGD, so a lot of prominent experts of this topic. However, they found, paradoxically, that the use of biologics against oral phototherapy, even no biologics, was associated with a higher risk of arthritis, paradoxically. And this was also confirmed by another study, always based on electronic health record database by Joe Merola, that showed that you see biologic here, and the risk of arthritis was higher than non-biologic standard treatment treated patients. So again, confirming this paradoxical association between use of biologics and development of arthritis. Another study recently published from electronic health records showing that TNF-alpha inhibitor was associated with a higher risk of PSA compared with IL-23 and even slightly compared with IL-17. So what's going on? If we prescribe a biologic or even specifically a TNF-alpha inhibitor, are we increasing the risk of PSA? Of course, no, I would say, because there is a fundamental biases there. And this is the commentary on the recent literature saying that when you perform a confrontation, a comparison between two groups treated with two therapies, of course, the group of patients you're treating is completely different. And these differences can eventually impact on the risk of PSA. If you don't measure every single covariate, every single variable, you might encounter this problem. So you have a lot of problem with observational data, as already mentioned. How do we deal with this? For instance, confounding by indication. So you might have different, let's say, PASI for different drugs. In your mind, you're using a powerful drug with a patient with a severe psoriasis. Of course, we all do this. But we know that PASI is associated with increased risk of PSA. The degree of activity of skin activity, let's say, is associated with this. So we need to address this. How do we do that? We control for these confounders. We need to collect data about everything: PASI, nail disease, inverse disease, history of psoriatic arthritis, and so on. And when you have all the covariates, you can match, let's say, these differences with a propensity score matching. You have other methods. This is something that a lot of studies have performed. Of course, there is still a problem with a measure. So when you don't measure some confounding factors, you might still find differences, even if you do a propensity score matching. So, for instance, in the literature, they didn't measure all of the studies: PASI, arthralgia, the presence of arthralgia, nail psoriasis, and so on, and BMI sometimes. So all factors able to impact on the development of PSA. And talking about development of PSA, there is something more in this topic. That is the protopathic biases. What is the protopathic bias? Is a bias that occur when you use a treatment in a patient that is affected by psoriasis, but is affected also from other diseases or other symptoms, and you use that drug because you think also about these other diseases or these other symptoms. So you're not only thinking about psoriasis, but for instance, you think about arthritis. When you have a patient with psoriasis and arthralgia, even if it doesn't have the label, let's say, diagnosis of arthritis by a rheumatologist, you might prescribe a TNF-alpha inhibitor instead of a IL-23 inhibitor. That would eventually impact on the development of arthritis itself in the future. So how to handle this? You need to do matching with propensity score, but you need to measure arthralgia. And none of the studies that I showed you in the last few slides are detecting arthralgia in their analysis. So in conclusion, of course, we've seen that real-world data are excellent, are exciting, and sometimes are really important, but they have caveat, they have biases, they have issues that need to be understood and well taken by all the readers of this paper. Thank you very much.